This lecture is on pentose phosphate pathway, also known as hexose monophosphate pathway, as well as phosphogluconate pathway. All three names are indicative of the intermediates found in the pathway, and it has been named in that manner. In terms of pentose phosphate pathway, if I am to give any exam question, I will refer it as simply as pentose phosphate pathway. The pentose phosphate pathway is in fact an alternative for the fate of glucose. Other than pentose phosphate pathway, which is involving in the conversion of substance of biological importance, that is the third bullet given here, the other fate of glucose includes oxidation. As you know, oxidation of glucose involves the glycolytic pathway, which eventually will lead to generation of ATP. It's also possible the excess of glucose is to be made into glycogen, that is through glycogen synthesis, with of course the crucial enzyme of glycogen synthase as the rate limiting enzyme. Apart from that, glucose can also be converted to acetyl coenzyme A by glycolytic pathway and can be then entering the fatty acid synthesis in order to form fatty acid. So these are all the fate of glucose, where one that will be of relevant to this lecture is the conversion to biological importance. So in this slide, you will look at the alternative of glycolysis, but in terms of two different organisms. One is prokaryote, while the other is eukaryote. For the prokaryote, since it's only a single cell organism, therefore it doesn't need anything very complex, and it only has one pathway doing all the requirement necessary to have formation of ATP, NADH as well as NADPH, and that's the Etner-Dodorov pathway as shown on the diagram on the right. Well, that of a eukaryote, much more complex with a multi-cell, hence require not only glycolysis. Glycolysis will produce NADH, ATP, but also require another pathway, that is pentose phosphate pathway, in order to generate NADPH, and that's been highlighted here. This slide basically summarizes in terms of the fate of glucose in any organism. Basically, glucose can be made into energy, ATP, by the glycolytic pathway. And if the excess is present, it can be then made into glycogen. Glycogen, of course, in animal cells, in particular liver as well muscle cells, there will be storage of glycogen. And then if it is a plant, it is possible to be made into starch or sucrose for storage purposes as well. In terms of biosynthesis, the pentose phosphate pathway plays the role where it can be oxidized into forming NADPH as well as ribose 5-phosphate. On a whole, pentose phosphate pathway is relevant in generating NADPH as well as the production of ribose. In terms of production of ribose, it is the residue relevant in making the sugar part or portion of nucleotide as well as nucleic acid, RNA or DNA synthesis. Then you have in terms of NADPH, it is a reductant. This NADPH is useful in many of the biosynthesis pathway, such as in the making of fatty acid, in the formation of cholesterol, one of the steroids, as well as in making amino acids. in particular one reaction that involving the enzyme glutamate dehydrogenase. It's also useful in terms of safeguarding the cell from highly reactive oxygen species with the regeneration of glutathione in red blood cell as well as other cells. And this pentose phosphate pathway, in terms of where it is active in most cells or tissues, except in non-lactating mammary gland, as well as very low activity in skeletal muscle. So here you can see all the three names given for this pentose phosphate pathway, also known as phosphogluconate pathway, also known as hexose monophosphate pathway. The it refers to the either the ribose sugar or the 6-carbon sugars present in the pentose phosphate pathway. This pathway can be separated into two, that is either the first phase, which is oxidative phase, or the second phase being non-oxidative phase. The oxidative phase mainly will produce NADPH in two reactions found there. And then you have the second phase, which will produce the ribose 5-phosphate. But apart from the 5-carbon sugar, it also generates other sugar molecules, which has 4-carbon, 10-carbon, 7-carbon, 6-carbon, as well as 3-carbon sugars. And then in terms of its location, it is in cytosol, and in terms of its whether can be categorized as catabolic or anabolic, it is mainly categorized as anabolic. However, it can also play a catabolic role, as you will see later on. So in terms of importance of this pathway, of course, mainly for the production of NADPH as well as ribose sugar. And if you want to compare which one is more important, obviously NADPH compared to ribose sugar. And then apart from NADPH serving as for biosynthesis pathways, it is also relevant for the protection of cell from the highly reactive oxygen species such as hydrogen peroxide. If there is anything going wrong in this pathway, as you will see, there will be a genetic mutation on one of the enzyme, the crucial enzyme, and that will lead to low production of NADPH, and in turn it could lead to anemia, that is hemolysis of red blood cell. This has already been covered, that is it is found primarily located in the cytoplasma and not found in the muscle as well as non-lactating mammary gland. So we cover one of the function of NADPH, that is one of the crucial function in eliminating highly reactive oxygen species within our body. In terms of where it is found in erythrocyte, pulmonary cells, as well as liver cells. Pulmonary is on the respiratory tract. So when there is much hydrogen peroxide in these three locations, what happens is glutathione in reduced form will be useful in reducing hydrogen peroxide to become something safe, that is water. In the process, however, reduced glutathione will become oxidized glutathione, that is forming the thiol bridge. And when that happens, the reduced glutathione will be level will be low, and in order to regenerate, this is where the NADPH will be utilized, where it will reconvert oxidized glutathione to become reduced glutathione again, which in turn useful in deactivating more hydrogen peroxide. Here, given the comparison between reduced glutathione on the left as well as the oxidized glutathione on the right. And in terms of glutathione, it is actually made up of tripeptide. So it is a protein made up of glutamic acid, cysteine, as well as glycine. And the one that is very relevant is the cysteine, where it takes part in the reaction itself. So glutathione is somewhat considered like a natural antioxidant system found within your body. So it has ability to remove all these very reactive oxygen species, not only hydrogen peroxide, as you can see also other example given here. There are two enzymes play a role, that is glutathione reductase as well as glutathione peroxidase, in this system of eliminating something which is highly reactive, where it can oxidize all the four macromolecule, either protein, lipid, carbohydrate, or nucleic acid. Quite detrimental if it's nucleic acid, where the nucleic acid can undergo mutation once it's oxidized, and that could even lead to eventual cancer. So just like any other biochemical pathway, this pentose phosphate pathway also broken down to phases. It's broken down to two phases, that is oxidative as well as non-oxidative. In terms of the whole pathway, it is relevant in generating NADPH as well as ribose 5-phosphate. The oxidative phase has the rate limiting step for the whole pathway itself. That is the first step, glucose 6-phosphate dehydrogenase enzyme. And then the oxidative phase has three steps that has two steps of it forming NADPH. The non-oxidative phase will generate the ribose 5-phosphate as the most important intermediate compared to the other intermediates, such as the 3-carbon glyceryl diphosphate, the 6-carbon fructose 6-phosphate, the 4-carbon erythrose 4-phosphate, the 7-carbon pseudoheptulose phosphate, and then you also have the 10-carbon xylulose 5-phosphate. And then you also have these enzymes, trans-ketolase, trans-aldolase, where they able to connect between glucose to glyceraldehyde 3-phosphate as well as fructose 6-phosphate. If you recall, remember, these intermediates such as glyceraldehyde 3-phosphate and fructose 6-phosphate are in fact intermediates of glycolytic pathway. So meaning this pentose phosphate pathway is in fact connected to a glycolytic pathway by these two intermediates. A bit detail on the steps, at least the three steps of all the steps in pentose phosphate pathway. The first step, as shown here, is the making of NADPH, where the crucial enzyme glucose 6-phosphate dehydrogenase is found. So basically the two hydrogen will be taken up by the NADP to become NADPH plus an hydrogen ion accompanying it. Also been shown here at the bottom, the enzyme is highly specific to NADP rather than NAD, since the Km is much lower, 1000 times lower than for the NAD. So that's why it prefers NADP. This is the second step. Not much to describe here. Let's move on to the following step. So this is the third step in the oxidative stage, and also the last step where it actually generates NADPH. So this step also shows that 6-phosphogluconate can be carboxylize in order to remove one carbon as carbon dioxide. In terms of the non-oxidative phase or stage, too many steps and too complex to actually go one at a time. So it's just been summarized in this manner. So obviously ribose 5-phosphate is the most important intermediate that need to be produced in this phase. And then also been highlighted in red the formation of glyceraldehyde 3-phosphate as well as fructose 6-phosphate, since it is intermediate that is connected to glycolytic pathway. And then the enzymes transketolase as well as transaldolase relevant in forming this glyceraldehyde 3-phosphate as well as the 6-carbon fructose 6-phosphate. So here you can see what I just described just now, that the 5-carbon ribose 5-phosphate can eventually, due to transketolase as well as transaldolase, can form the 3-carbon glyceraldehyde 3-phosphate as well as 6-carbon fructose 6-phosphate. So because of these changes, it is then possible to link between glycolytic pathway as well as pentose phosphate pathway. So it's been referred, the pentose phosphate pathway is inactive in the muscle, and that's due to the fact that there is inactivity found in two of its enzymes, that is glucose 6-phosphate dehydrogenase as well as 6-gluconate dehydrogenase. So because of inactivity of these two enzymes, therefore it is inactive in the muscle, but it's not possible to form NADPH. It's still possible to form ribose 5-phosphate by the reverse action from the glycolytic pathway, forming the fructose 6-phosphate as well as glyceraldehyde 3-phosphate, and then eventually can form ribose 5-phosphate. So in terms of the regulation of this pathway, it is by regulating the crucial enzyme, the rate limiting step, that is catalyzed by glucose 6-phosphate dehydrogenase, which happen to be the first step of this pathway itself. And how it's done is by allosteric regulation, where its own product, that is NADPH, when become excessive in amount, it will then bind to the allosteric site and cause a conformational change to the enzyme, and therefore inhibiting it. It's also possible by covalent modification. Usually hormonal regulation is by covalent modification, and hence insulin, once is secreted, it will cause this enzyme to be activated by covalent modification and therefore active to do its function. So here we're going to look into the pentose phosphate pathway, how it safeguards the red blood cell or erythrocyte. Things which are producing highly reactive oxygen species, such as taking certain drugs, drugs which are taken on a daily basis because of prescription, may cause increase in highly reactive oxygen species, as well as certain microbial infection. And another that's been identified is the consumption of a particular kind of food, that is roasted fava beans, as shown in the picture there. So all these issues will create highly reactive species such as that of hydrogen peroxide. When that happens, the hydrogen peroxide need to be eliminated from the erythrocyte. If not, it will cause hemolysis of the red blood cell, and that will be a problem. So how the erythrocyte does that to eliminate the hydrogen peroxide is by converting it to water. with the use of reduced glutathione. However, as already been referred to you earlier, the reduced glutathione also will undergo changes and it will become oxidized glutathione. In order to reform reduced glutathione, it requires the reductant NADPH to oxidize it, to reduce it back to reduced glutathione. And this NADPH is produced by the pentose phosphate pathway that takes place in the erythrocyte. Just want to bring your attention to the left of the erythrocyte where shown glycolytic pathway, where it does not form all the way to become carbon dioxide and water, simply because the erythrocyte is missing mitochondria. So it only forms up to lactate. And this lactic acid is also very acidic, therefore it needs to be transported to the liver, where it can be through the gluconeogenesis pathway can be made into glucose. So here we look at what happens if there is a genetic disorder relevant to glucose 6-phosphate dehydrogenase. This genetic disorder takes place in the X chromosome. So that means the mother able to bring it down, the defective gene, to almost all the sons, because Y chromosome does not have any gene whatsoever. So that means if the X chromosome has the defective gene, therefore the sons are normally affected in bringing down the defective gene. And if the defective gene is found, the glucose 6-phosphate dehydrogenase will be deficient and inactive. Therefore, it cannot produce NADPH through pentose phosphate pathway. And therefore, if highly reactive oxygen species is found, such as hydrogen peroxide, it will cause oxidation of the erythrocyte or red blood cell. And eventually, lysis of red blood cell will be found, and this is through hemolysis. And then the content of the red blood cell, which is bilirubin, will be then thrown out of the hemolyzed red blood cell, and it will be then deposited on tissues, because too many to be detoxified by the liver. So what happens is it will then deposits on certain tissues such as the skin as well as the eye, and it will start to look like yellow, and that's where the symptom of jaundice takes place. So this is the one I described earlier about all the males to be affected because the gene, defective gene, is found in the X chromosome. Also been shown here is the second, also the third bullet, where it also says that this jaundice symptom also takes place in newborns. You see, when babies are born, some of the internal part is not actually fully formed before coming out to the world. So one of it is sometimes glucose 6-phosphate dehydrogenase is not adequately, sufficiently created. So what happens is it will cause deposit of bilirubin, and the baby will start to look yellow, jaundice. So in order to treat the baby, what happens is the hospital will keep the baby longer and exposed to certain light, which will then break down this bilirubin, so unable to be released from once born immediately. So here I give you the summary of the pentose phosphate pathway, where its function not only mainly for the production of NADPH as well as converting hexose to pentoses, but also, although not the main function, is also to perform complete oxidative degradation of pentoses, where the 5 carbon sugar can also enter glycolytic pathway via pentose phosphate pathway once it forms the fructose 6 phosphate as well as the glyceraldehyde 3 phosphate. And also the NADPH, which is the main function of pentose phosphate pathway, it has many functions. This reductant is able to be used in several biosynthetic reactions such as fatty acid synthesis, amino acid synthesis, as well as cholesterol synthesis. You also have the NADPH serving as a biochemical reductant for the removal of highly reactive oxygen species. You also have this NADPH quite required in the liver cell, where this P450 monooxygenase system is found in order to react with xenobiotics such as alcohol consumption, excessive alcohol, as well as certain degradation of drugs. And then it's also useful in converting the ribose sugar to deoxy ribose sugar of nucleotide, where the C2 prime carbon is removed of the OH to be added with hydrogen instead. So these are all the function of NADPH. In terms of conversion of hexose into pentoses, of course, apart from NADPH, it also makes ribose 5-phosphate, and this is used in forming the component of ATP as well as other coenzymes, the sugar part of these coenzymes itself, as well as nucleic acid.